If you break your arm, the X-ray shows this clean, jagged, white line, right? And the diagnosis is just incredibly binary. It's broken or it's not.
Right, exactly. But when you step into the world of cancer therapy, that satisfying clarity just completely vanishes.
Yeah, it really does. We're navigating a diagnostic and treatment landscape that is just wildly unpredictable, especially, when we look at the supposed Trojan horses of modern oncology, which are antibody-drug conjugates or ADCs.
Oh, absolutely, the ADCs.
Yeah, so today we're doing a deep dive into a stack of recent oncology papers and molecular mapping studies. We're unpacking a biological mystery that is honestly completely upending how we understand tumor resistance.
And, the clinical context for this mystery really centers on a specific drug called Enfortumab vedotin.
Right.
It's an ADC specifically engineered to target a protein called Nectin-4. And if you look at the clinical trials over the last few years, specifically, the EV301 through EV304 trials, this drug has been hailed as a massive breakthrough.
A total game changer for urothelial carcinoma, right? Which is basically muscle-invasive bladder cancer.
Exactly. Muscle-invasive bladder cancer.
Because conceptually, an ADC is just a brilliant piece of biological engineering. You take a monoclonal antibody that's designed to seek out a highly specific protein on the surface of a tumor cell, so our Nectin-4. Then you attach this really highly toxic chemotherapy payload to that antibody. So the antibody finds the target, binds to it, and then the cancer cell literally engulfs the entire complex.
Right, it pulls it inside.
Yeah, and the TOPS payload is released internally, so it basically decimates the cancer cell from the inside out while, largely sparing the surrounding healthy tissue. It's like an elegant guided missile.
It is, yeah. But the data shows a pretty significant failure rate.
Right, and that's the dark side of this breakthrough.
Yeah, roughly 40% of patients either exhibit primary resistance, meaning, the drug never works for them at all.
Wow, 40%.
Yeah, or they experience acquired resistance, where the tumor initially shrinks, but inevitably comes roaring back.
Which brings us to the core mystery of the sources we are looking at for you today. Because logically, right, if a tumor develops resistance to a targeted therapy, you'd assume it simply stopped expressing the target.
Right, that's the classic assumption.
It recognized that the Nectin-4 protein was a liability, like a glowing bullseye, and it downregulated that protein so the guided missile has nothing to lock onto.
And that evasion tactic is, it's what we've historically expected with target therapies. You hit the target, the tumor mutates to hide it.
Makes total sense.
Right. But when researchers actually looked at the molecular profile of these resistant bladder tumors, they found the exact opposite.
Wait, the opposite?
Yeah. They uncovered a highly resilient tumor state where the expression of Nectin-4 on the cell surface was actually significantly higher than it was before the treatment started.
See, that is entirely paradoxical. How does a tumor survive a therapy designed to kill it through a specific target while simultaneously painting a massive intensified bullseye on its own back?
It's a massive plot twist.
It really is. And that is the mission for this deep dive. We are going to dissect the mechanism of this resistance down to the molecular level and look at a radical new therapeutic strategy designed to just completely outsmart it.
So to understand this mechanism, we really have to look at how researchers mapped the ecosystem of the resistant tumor.
Right, the scene of the crime.
Exactly.
Yeah.
They built this highly controlled preclinical model. They took treatment-naive human muscle-invasive bladder cancer and engrafted it into a specialized mouse model, specifically the UCD34 plus HSC-NCG strain.
Okay, let me jump in here because for those of you listening, the humanized aspect of this mouse model is just absolutely critical.
That's the whole point.
Right. We aren't just looking at human tumor cells growing in a random mouse. These mice have been engrafted with human hematopoietic stem cells. They actually possess a functioning human immune system, complete with human T cells and macrophages.
Because you can't just study a tumor in a vacuum.
Exactly. It exists in this dynamic neighborhood called the tumor immune microenvironment. If you want a medically relevant simulation of how a tumor reacts to an ADC, you need the human immune system actively patrolling that microenvironment.
Yeah, and the presence of that immune system makes the subsequent data just far more robust.
Yeah.
So researchers treated these humanized mice with the Nectin-4 ADC and then applied single-cell RNA sequencing to compare the pre-treatment tumors with the resistant tumors.
The ones that actually survived the therapy.
Right. And when they analyzed the resistant group at a single-cell resolution, this massive anomaly emerged. a specific subpopulation of tumor cells, which they designated as the C1 Nectin-4 positive cluster, had completely taken over.
And let me guess, this specific cluster exhibited the highest levels of Nectin-4 expression.
The highest of any cell type in the study.
See, I find myself inherently skeptical of mouse models, though. I mean, no matter how sophisticated the humanization process is, the leap from a murine model to human oncology is historically fraught.
Totally fair point.
Right. So are we seeing this exact same C1 Ection-4 positive dominance in actual human patients who have failed Enfortumab vedotin therapy?
We actually are. The translation to human patients is definitively confirmed in the data. The researchers utilized paired spatial transcriptomics, specifically the StereoSec platform on tissue samples from 6 human patients.
Okay, spatial transcriptomics is such a powerful tool because, it gives you the actual geographic coordinates of gene expression. You aren't just seeing what genes are turned on.
Right, you're seeing exactly where in the architecture of the tumor they are active.
Exactly. And these were patients with metastatic urothelial carcinoma, right?
Yes. So they had lesions across the bladder, the renal pelvis, lymph nodes, liver. And the analyzed biopsies taken both before and after the patients received the Nectin-4 ADC, along with an anti-PD-1 immunotherapy.
Okay, so to process that spatial geographic data, what did they use?
They applied non-negative matrix factorization, or NMF. And through that NMF analysis, they identified a distinct genetic signature they called metaprogram 3, or MP3.
And this MP3 signature basically represents that Nectin-4 high tumor state.
Exactly. And the spatial data unequivocally showed that this MP3 state was massively enriched in the post-treatment resistant human lesions.
Wow.
Yeah, the spatial distribution in the human patients perfectly mirrored the single-cell data from the mouse models.
Okay, so the target is undeniably there. The antibody is successfully finding it and binding to it. But an ADC only works if the toxic payload actually enters the cell.
Right, it has to get inside.
And the chemical payload in this specific drug is MME, right? Monomethyl auristatin E. So what did the mass spectrometry data actually show about the intracellular concentration of MMAE?
Well, the LC-MSMS data revealed the ultimate bottleneck. Despite the overwhelming presence of the Nectin-4 target on the surface, and despite the drug binding to those targets,
Toxic MMAE payload wasn't getting in.
it was barely accumulating inside the resistant cells at all.
That is wild. It's the equivalent of having a house entirely covered in mailboxes. The mail carrier arrives, clearly sees the address, walks up to the porch, but is somehow physically prevented from placing the mail inside any of the boxes.
That's a perfect analogy.
The letters, the toxic payload, are just piling up on the front porch. Okay, let me pause here to make sure we have the architecture of this straight in our heads. This brings us to our first 5-minute recap.
Sounds good.
Okay, so for you listening, here is the summary of what we've uncovered so far. Bullet 1. ADCs are targeted Trojan horses. Bullet 2. Urothelial cancers develop resistance not by deleting the target, but by amplifying it, creating this Nectin-4 high state. And bullet 3. The toxic MMAE payload isn't getting inside the cell.
It's locked out.
So here's an interactive question for you to chew on. If you were the tumor and you couldn't get rid of your surface targets, how would you physically stop the drug from coming through the front door?
Which is the immediate mechanical question we have to answer. If the target is present and functioning, what is the physical mechanism the cell is using to board up the door and prevent the drug complex from entering?
Right. To understand the blockade, we first need to understand how the door operates when it isn't boarded up. Like, how does a Nectin-4 protein normally transport something from the outside of the cell to the inside?
Well, the standard internalization pathway is clathrin-mediated endocytosis, or CME. You can picture clathrin as this geometric molecular cage that assembles on the interior wall of the cell membrane.
Okay, I can see that.
It physically pulls the membrane inward to create a vesicle, essentially swallowing whatever is bound to the exterior surface.
But the clathrin cage doesn't know what to grab on its own, does it?
No, it doesn't. It requires a highly specific adapter protein called AP2M1 to act as a molecular doorman.
Okay, so this doorman, AP2M1, is stationed on the inside of the cell, scanning the intracellular tails of transmembrane proteins. What specific structural motif is it looking for to trigger that clathrin machinery?
So AP2M1, recognizes a highly conserved amino acid sequence known as a canonical YXX5 motif.
YXX5.
Yeah. On the intracellular tail of the Nectin-4 protein, this motif consists of 4 specific amino acids: tyrosine, serine, threonine, and leucine. So YSTL.
YSTL.
Exactly. This sequence sits exactly at positions 445 through 448 on the protein chain. When AP2M1 binds to that YSTL sequence, it initiates the endocytosis process, and the ADC is pulled inside.
But in our resistant C1 and Nectin-4 positive cells, the door isn't opening. The researchers ran affinity purification mass spectrometry, or APMS, to fish out whatever proteins were interacting with the intracellular domain of Nectin-4 in these resistant cells.
And they caught a massive outlier.
Right. The analysis revealed staggering concentrations of a protein called AKR1C1 bound to the Nectin-4 tail.
Which is bizarre.
Yeah.
Because under normal biological conditions, AKR1C1 is an aldoketoreductase enzyme. It's typically involved in processing steroid hormones and detoxifying various compounds.
It's an enzyme. It's a chemical processing unit. But the data suggests it isn't doing any chemical processing here. It has completely abandoned its day job to moonlight as a structural barricade.
The molecular docking simulations confirm this exact structural role. The simulations demonstrated that the catalytic pocket of AKR1C1, the physical groove it normally uses to bind and process toxins, fits flawlessly over the intracellular domain of Nectin-4.
Wow. Where exactly?
Specifically, it binds across amino acids 443 through 472.
Ah, that binding footprint is the smoking den. Positions 443 to 472 completely swallows the YSTL sequence at positions 445 to 448.
It covers it entirely.
So if AP2M1 is the key required to unlock the Clathrin machinery and the YSTL sequence is the keyhole, the tumor cell has taken this AKR1C1 enzyme and jammed it perfectly over the keyhole like a massive wad of chewing gum.
I love that visual. And the structural interference is absolute. The binding of AKR1C1 and AP2M1 to that region is mutually exclusive.
They can't both be there.
Right. By maintaining high levels of AKR1C1, the tuner ensures that the AP2M1 adapter can never access the YSTL motif. The Clathrin cage never assembles, the endocytosis pathway remains entirely dormant, and the highly toxic ADC is left stranded on the exterior surface of the cell membrane.
Okay, so let's do our second recap here.
All right, listeners, here is the summary of the blockade. Bullet 1, endocytosis requires the AP2M1 protein to bind to the YSTL motif on Nectin-4. Bullet 2, in resistant cells, AKR1C1 physically covers this motif. And bullet 3, the ADC is completely locked out of the cell.
Which leads to our next interactive question for you. Okay, so the door is blocked, but these ADCs are still lashing onto the outside of the cell. They can't just pile up on the surface forever without causing damage. So how does the tumor take out the trash?
Yeah, that's the big mechanical problem.
Right, because I was losing the thread a bit there. If the cell is constantly producing more Nectin-4 targets and the drug is constantly binding to them, that exterior membrane is going to get incredibly crowded.
Very crowded.
And a cell can't just accumulate infinite amounts of an antibody-drug complex on its surface without the lipid bilayer destabilizing. The mechanical stress alone would trigger apoptotic pathways, and the cell would just die. How is the tumor not collapsing under the sheer physical weight of this blockade?
Well, the preservation of membrane dynamics is critical for cell survival. The tumor can't just block the door, right? It has to have a secondary mechanism to clear the accumulation.
Needs a garbage chute.
Exactly. And the data reviews that AKR1C1 is actually functioning as a double agent. It doesn't merely act as a deadbolt on the door. It actively coordinates a waste disposal system.
So it's setting up an alternative turnover route. How did the researchers physically observe this happening? Because, I mean, you can't exactly look at a cell through a standard microscope and see individual proteins being disposed of.
No, you really can't. So they analyzed the culture supernatant, which is the fluid immediately surrounding the resistant cells. They utilized ultracentrifugation, basically spinning the fluid at immense speeds to isolate extracellular vesicles, or EVs.
These are those microscopic lipid bubbles that cells naturally shed into their environment.
Right, and once they isolated them, they employed immunoelectron microscopy. They tagged the Nectin-4 and AKR1C1 proteins with nanoscopic gold particles to visualize their exact locations at extremely high resolution.
Oh, wow. And the gold particles mapped exactly to those extracellular vesicles. So the Nectin-4 ADC complexes weren't just sitting statically on the cell surface.
Not at all.
They were being aggressively jettisoned, packaged inside these shed vesicles, and floating harmlessly away from the tumor cell.
And the structural biology behind how AKR1C1 orchestrates this shedding is just remarkable. While the catalytic pocket of AKR1C1 is bound to the Nectin-4 tail, Another specific region of the AKR1C1 protein remains exposed to the interior of the cell.
Okay, so what's on that exposed region?
It contains a PPNY motif: proline, proline, asparagine, and tyrosine, at residues 314 to 317.
So while the catalytic pocket is blocking the endocytosis keyhole, this PPNY motif is facing inward, acting as a molecular beacon. What exactly is the beacon signaling for?
Well, the PPNY motif has a very high affinity for a specific E3 ubiquitin ligases called WWP2.
WWP2, the cleanup crew.
Exactly. And the binding of WWP2 serves as the catalyst for the entire waste disposal process.
Yeah.
Because once WWP2 is recruited to the site, it activates a sophisticated piece of cellular machinery known as the ESCRT complex.
Okay, if you visualize the cell membrane as a glass blower's bubble, the ESCRT complex is the highly specialized tool that pushes a small section of that bubble outward, tightly pinches the neck, and snips it off to create a completely separate, free-floating mini-bubble.
That's precisely what it does.
The ESCRT complex effectively bags up the toxic ADC stuck on the surface and throws it out the cellular window.
Yep, and the experimental validation for this pathway is definitive. When the researchers genetically deleted WWP2 from the resistant cells, the entire shedding mechanism collapsed.
Let me guess, the EVs weren't formed.
No EVs. The ESCRT complex was never activated, and the ADC complexes immediately began accumulating to toxic levels on the cell membrane again.
Wow. Okay, time for our third recap. For you listening, here's what we just learned about the escape route. Bullet 1. AKR1C1 blocks the internalization of the ADC. Bullet 2. AKR1C1 uses its PPNY motif to summon WWP2. And bullet 3. WWP2 triggers the ESCRT complex to shed the ADC in extracellular vesicles.
It's quite the operation.
It really is. So here's your question to think about. We now know the mechanic, which is AKR1C1, and the garbage truck driver, WWP2, that are keeping the tumor alive. But who is the mob boss giving the orders to hire them in the first place?
Right, because proteins don't just spontaneously decide to restructure a cell's defenses. There has to be a higher-level genetic architect issuing the blueprints for this entire resistant state.
So how did they find the architect?
To identify the upstream architect, the research team deployed CNIC. It's a computational algorithm designed to infer complex gene regulatory networks from single-cell data.
Okay.
And they cross-reference those computational findings with GIP assays, chromate immunoprecipitation.
Right. GIP allows researchers to map exactly where regulatory proteins are physically binding to the DNA helix. So they were hunting for the specific transcription factor responsible for flipping the switches on this whole resistance program.
Exactly. And the GIP assays isolated a transcription factor called ELF3. The data proved that ELF3 physically occupies the promoter regions, the genetic starting blocks, of both the Nectin-4 gene and the AKR1C1 genes simultaneously.
So ELF3 is the master switch driving the co-expression of the target and the blockade.
It's the mob boss.
Okay, but from a purely strategic standpoint, if ELF3 is the master switch coordinating the entire resistance program, the clinical solution seems kind of obvious. We just develop a drug to inhibit ELF3, right? You cut the head off the snake, and the entire C1 and Nectin-4 positive state collapses. Why isn't that the primary therapeutic goal?
Well, targeting transcription factors is notoriously viewed as a pharmacological graveyard.
A graveyard, really?
Yeah. Structurally, transcription factors like ELF3 often lack the deep, accessible binding pockets that small molecule drugs require to latch on effectively. Their surfaces are relatively flat. Plus, they operate deep within the nucleus.
Making them hard to reach and grip.
Exactly. But more importantly, ELF3 is not exclusive to this resistance mechanism. It plays a foundational role in the normal differentiation and homeostasis of healthy epithelial cells all throughout the body. Systemically drugging ELF3 would likely result in catastrophic off-target toxicity.
Got it. It's a systemic biological load-bearing wall. You can't just knock it down without collapsing the healthy tissue right alongside the tumor.
Precisely.
Which means we have to bypass the architect and go after the middleman executing the blueprints. The structural vulnerability is AKR1C1.
And the therapeutic pivot to targeting AKR1C1 is where this research moves from just a fascinating biological observation to a truly viable clinical strategy. The researchers hypothesize that if they could occupy the catalytic pocket of AKR1C1 with a competitive inhibitor, it would physically prevent the protein from clamping down on the Nectin-4 tail.
You essentially need a chemical solvent to dissolve the chewing gum blocking the keyhole.
That's it, exactly. And the initial screening identified a competitive inhibitor called CPSA. It successfully disrupted the interaction, but animal models revealed severe hepatotoxicity and nephrotoxicity, so liver and kidney damage.
Not ideal.
No. However, they identified an alternative compound, 5-PBSA, which demonstrated the exact same ability to disrupt the AKR1C1 Nectin-4 interaction, but possessed a highly favorable safety profile.
Okay, this is the moment of truth for the entire mechanistic model. What happens to the resistant humanized mouse models when you administer the Nectin-4 ADC in combination with the 5-PBSA inhibitor?
The reversal of resistance was complete. The 5-PBSA molecule bound tightly to the AKR1C1 catalytic pocket, physically forcing it to detach from the intracellular domain of Nectin-4.
Wow.
With the blockade removed, the YSTL motif was suddenly exposed again. The AP2M1 adapter protein immediately recognized the motif, successfully triggered clathrin-mediated endocytosis, and the cancer cell forcefully engulfed the targeted ADC.
The Trojan horse finally got past the gates.
Exactly. The intracellular accumulation of the toxic MMAE payload was entirely restored. And because AKR1C1 was displaced, the recruitment of WWP2 and the subsequent vesicular shedding completely ceased.
It's just a brilliant pharmacological workaround. But knowing researchers, they pushed the envelope even further, didn't they? They didn't just look at the intracellular mechanics. They look at how restoring this pathway impacted the broader tumor immune microenvironment.
Oh, they did. The final and arguably most compelling data comes from a triplet therapy model. They treated the humanized mice with the combination of the Nectin-4 ADC, the 5-PBSA inhibitor, and an anti-PD-1 immunotherapy.
Okay. Anti-PD-1 therapies are checkpoint inhibitors. They essentially strip the molecular camouflage off the tumor and take the biochemical brakes off the patient's own CD8 positive T cells, allowing the immune system to recognize and attack the cancer. But they often struggle if the tumor microenvironment is immunosuppressive.
Right, but the synergy of the triplet therapy transformed that microenvironment completely. By using 5-PBSA to force the cancer cells to ingest the ADC, the tumor cells underwent massive internal apoptosis. As they died, they released a flood of tumor-associated neoantigens and pro-inflammatory signals.
Alerting the immune system.
Yes. When researchers analyzed the tissue using flow cytometry, the results were definitive. The microenvironment, previously cold and resistant, was suddenly saturated with highly active, cytotoxic CD8-positive T cells.
Wow.
Concurrently, the population of tumor-associated macrophages, or TAMs, which typically suppress the immune response, precipitously declined. The combination resulted in profound, durable tumor regression.
Okay, time for our final recap.
All right, listeners, here is the big payoff. Bullet 1. ELF3 is the master switch driving this whole resistant state. Bullet 2. Inhibiting AKR1C1 with 5-PBSA restores ADC uptake and stops vesicle shedding. And bullet 3, combining this with anti-PD-1 creates a powerful immune synergy that defeats the resistant tumor.
So to synthesize the journey we've been on today, what began as an immense clinical frustration, patients failing therapy despite overwhelming evidence that the drug's target was present, has been decoded into this astoundingly complex molecular choreography.
It really is beautiful in a dark way.
It is. We have a master architect, ELF3, driving an overexpression of a target. We have a hijacked enzyme, AKR1C1, barricading the AP2M1 endocytosis gateway. And we have a coordinated waste disposal system utilizing WWP2 and the ESCRT complex to jettison the toxic threat in extracellular vesicles.
It's a profound testament of the sheer evolutionary willpower of a cancer cell fighting for survival.
Absolutely.
And, the elegance of the resistance mechanism is undeniable. But the implications of this discovery extend far beyond this single drug or this specific bladder cancer.
Oh, for sure.
Because if a tumor can successfully coordinate membrane budding and exocytosis to physically shed this particular ADC, we have to deeply consider whether this target-high, uptake, defective vesicular shedding is the unrecognized culprit behind the failure of other advanced therapies.
That implication is just massive. It fundamentally challenges how we view resistance across the board. I mean, if you look at CAR T cell therapies, they rely entirely on latching onto surface antigens to initiate their cytotoxic effects.
Right.
Other highly successful ADCs used in breast cancer or lung cancer operate on similar internalization mechanics. If various tumor types are systematically utilizing the ESCRT complex to simply shed our most advanced targeted therapies in microscopic bubbles before the drugs can even execute their function, well, our current diagnostic tools just aren't equipped to see it.
Because the presence of a target does not guarantee the permeability of the cell. If we are going to rely on targeted therapies, we have to look past the surface expression and interrogate the mechanical integrity of the cellular entryways and the vesicular shedding pathways.
Yeah. That clear binary X-ray we talked about at the very beginning of the deep dive, it turns out it wasn't just broken. It was fundamentally the wrong tool for the job. It was designed to look for missing targets, completely blind to the fact that the tumor was quietly building a trapdoor to throw our most sophisticated weapons right back out.
Exactly.
Well, thank you for joining us on this deep dive into the resilient, beautifully complex, and honestly endlessly shape-shifting world of cellular biology. Keep questioning the obvious, and we will catch you on the next deep dive.